Geological Survey. It'll be January the 11th. Ren Starling will be talking about supermassive black holes in the center of our galaxy. And today I'll be introducing Professor Adi Radian from Civil Engineering at the Technion. She started her academic career at Shenkar in chemical engineering. She went on to do her PhD at the Faculty of Agriculture in Rehovot. She did a postdoc at Minnesota, and she's now been at the Technion since 2016. So we'll hear about...
Some years.
Yep, great, fantastic.
Thank you for that introduction. I'm very happy to be here today. So, as you said, I'm from the Civil and Environmental Engineering faculty at the Technion. I'm head of the Soil and Environmental Chemistry Lab, which I started about almost 10 years ago. And what I'm going to be showing today is a little bit of kind of an overview of the work that we've been doing. I'll start with our overall thinking. So we work a lot on remediation strategies for polluted water and polluted soils, and kind of the thing that we try to think about is that even though separation processes, which are very prevalent, absorption by activated carbon or membrane separation, they're very effective and they can be also very cost effective if they're done right, but they still just mainly move the problem from one phase to another. And in our thinking, we try to develop platforms that are more comprehensive, that look at transferring the pollutants from one phase to another, but also degrading them, destroying them. So what I'm going to be showing today is kind of two paths that we like to take. One that deals with absorption and degradation in a more chemical way, how we can create platforms that separate or absorb pollutants and then degrade them on the surface chemically. And if I have time, then I'll talk about our second strategy, which is absorption and biodegradation of different pollutants. So I'm not sure what your backgrounds are. If something that I say isn't clear, please feel free to stop me, because it's more important that you understand than I get through all the material. So our main materials that we work with are clay minerals and oxides. We try to use benign, naturally available materials as our basis for all the different platforms that we develop. Some of the work that we do in the lab just has to do with understanding the fate of different pollutants in the environment when we talk about their interactions with these type of minerals, and from there we try to understand if we can use them and how we can use them. The reason that we use these very benign, available materials is that they are low cost, they are non-toxic, and they have some intrinsic chemical and physical properties that can be very advantageous when we talk about remediation properties. So first of all, we use clay minerals that have a very high specific surface area. I always use maltmorillonite as an example. It's also the clay that we use most. It's a clay mineral that has a very high specific surface area. If you take 1 gram of clay and you spread it out, it will cover about 2 basketball courts. So 800 meters squared per 1 gram of clay. Very high specific surface area. These clays have a high cation exchange capacity, which means that they have a negative charge that can be compensated by cations, and that helps us play around and modify their surface, as we will see. And they're inert to oxidation processes. So when I talk about chemical destruction of pollutants, I mostly will be talking about oxidizing them. And so if I have a platform that's inert to that oxidation, then I can use it as just that platform, that basis, without hurting it, without degrading that platform itself. We also use oxides, mainly iron oxides and manganese oxides. Unlike clay minerals, they are very redox reactive. So when we talk about redox processes, they can be also very advantageous. So we really try to stay within the realm of these very benign and non-toxic materials. So this brings me to the first kind of path that I'm going to talk about, and that is designing catalysts for heterogeneous Fenton processes. Fenton catalysis is an advanced oxidation process used for remediating polluted water, where we take divalent iron ions and hydrogen peroxide. That activates the hydrogen peroxide to create hydroxyl radicals, and those hydroxyl radicals can attack organic pollutants and break them down. It's a very effective process and very rapid, but it's first of all, not very specific. Hydroxyl radicals will attack any organic matter, whether it's what we're targeting, or natural organic matter, or bacteria. And the second thing is that usually you need very low pHs to do this kind of process. Around pH 2.5 to 3 is the optimal pH for keeping the iron divalent and soluble, and for producing these hydroxyl radicals. And the last problem is that you create high quantities of iron oxide sludge when you use this process. So people have been moving away from classic Fenton reactions to heterogeneous Fenton reactions, which is what we use as well, where the catalyst is a solid catalyst. The iron is a solid, and this helps us separate the catalyst after it's done and we use it. It also allows us to work at a wide range of pHs, so we're no longer restricted to just very low pHs. The problem here is that it slows down the reactivity and the process vastly. You can see here the different kinetic constants for the two reactions. This is for divalent iron, this is for trivalent iron. So 3 to 4 orders of magnitude slower when we have a iron oxide that's oxidized. And the last thing is that we create a lot of reactive oxygen species. So these reactive oxygen species, they're not just hydroxyl radicals, so it gives us a larger toolbox on the one hand. On the other hand, it makes the process a lot more complex. So we have a slower process that's a lot more complex, and it's still not specific. So we wanted to build on a lot of the research that was done before and try to design catalysts that are more specific and that will still be fast and efficient. And the idea was to use the clay mineral montmorillonite as a platform. On that clay mineral, we deposit nano-sized iron oxides. Their small size allows for higher surface area and higher reactivity. And the large surface area of the clay is a good supporting platform, which will allow for a large area where these catalytic processes can actually happen. And we started out by looking at different iron oxides on the clay. You can see here we used hematite and magnetite and amorphous iron oxide. And we deposited these nano crystals on the clay mineral. We do this through wet deposition, so just using iron, and we deposit it, and then we change the temperature or we change the pH. Very low tech in terms of how we create this clay. And then we wanted to test and see whether this is actually a good heterogeneous Fenton catalyst. And for that, we used a model pollutant, phenanthrene, which is a polycyclic aromatic hydrocarbon, which is found in partial combustion, so you'll find it in polluted soils or polluted water from fuel products, or when we smoke, I don't smoke, but if you smoke, this is part of the pollutants that you inhale. So we use this as a model pollutant, and what you see here are two. Do we have a pointer? I think I saw. Does this work? No, probably not. Anyway. Yeah, I didn't put the USB in.
No,
so
just turn it on. Should be on the side of it.
Side.
Should be like a
Yeah,
slide.
I see. Oh, there we go. There we go. Okay. So we have two graphs here, and you can see here the degradation of phenanthrene and the different type of composites that we created, and we have the decomposition of hydrogen peroxide on the second graph. So this is the amorphous iron oxide, and then we have the other types of oxides here, and you can see that the amorphous iron oxide had the highest degradation ability towards phenanthrene, and in terms of how much hydrogen peroxide was decomposed, you can see that it wasn't the composite that initiated the most decomposition of hydrogen peroxide. So first of all, we saw that there wasn't a direct correlation between how much hydrogen peroxide is being used to how much phenanthrene is being degraded. That was interesting. So some of the composites will use up a lot of hydrogen peroxide but not degrade the phenanthrene at all. So this was interesting. And the amorphous iron oxides seem to have the best activity. Amorphous meaning it's not a specific crystal, right? So it has more defects and more types of iron species. So we kept looking at this catalyst. So this is now all with the iron, so amorphous iron oxide on the montmorillonite clay. You can see here the rate of phenanthrene degradation over time and the decomposition of hydrogen peroxide, which is consistent with what we saw before. Small amounts of hydrogen peroxide, high decomposition or degradation of phenanthrene. We looked for byproducts, we couldn't find any. This is an EEM of phenanthrene over time. You can see that there's just a decrease in the peak intensity, not more peaks coming up, and we couldn't identify anything in an LCMS or a GCMS analysis, so we couldn't find any byproducts coming up. And this made us wonder what the mechanism actually is, and we started looking at the different radicals that are formed. So we found two types of radicals. One is the hydroxyl radical that's being formed, and the other one is the singlet oxygen here. No superoxide, which is also sometimes formed during heterogeneous Fenton oxidation. So two types of radicals. And then we can use specific quenchers to take out an activity of one radical or another. So when I use sodium azide, if I add sodium azide, I'm canceling out the activity of the singlet oxygen. When I add tilbutyl alcohol, then I cancel out the activity of the hydroxyl radical. So I can pinpoint which radical is doing the work. And you can see here that when I add the sodium azide, which quenches the singlet oxygen, then I don't get any degradation of phenanthrene, or the degradation decreases. Whereas if I quench the hydroxyl radical, it's almost the same as if I don't add any quenchers. So we can find out that so we found out that the singlet oxygen is more potent than the hydroxyl radical for degradation of phenanthrene. And another interesting thing that we found was that we can pre-adsorb hydrogen peroxide and it stays stable until we add the phenanthrene, which is interesting. So in this experiment here, you can see cycles where we, in the first cycle, added hydrogen peroxide and phenanthrene into our system with the catalyst, and we looked at the degradation over time, or the decomposition of hydrogen peroxide. And you can see that we have 80% degradation and about almost 20% decomposition of hydrogen peroxide. In the next cycles, we just added phenanthrene, no more hydrogen peroxide, and we still get very nice degradation of phenanthrene and a linear increase in the amount of hydrogen peroxide that's being used up. So we can add hydrogen peroxide, it stays stable until we add phenanthrene, which made us think that the mechanism is that you have kind of an absorption or stabilization of hydrogen peroxide on the surface, and when phenanthrene is added, then the hydrogen peroxide is activated, and we get the formation of the radicals. And the reason we're not seeing, we think, the reason that we're not seeing any byproducts is because the rate limiting step is the diffusion to the surface. And then the degradation happens very quickly, so you don't see any byproducts. So that was the initial thought, and we kept kind of working on this catalyst, and we had an idea that, that maybe we can other way around. Maybe we can enhance the rate of degradation if we reduce the iron on the surface. Remember I showed you that reduced iron is much faster in activating hydrogen peroxide than the trivalent iron. And so we have this idea that we can reduce iron on the surface if we add specific compounds that have the ability to reduce iron in solution. So we worked with gallic acid, which is a catechol. Catechols are aromatic rings that have two adjacent hydroxyl groups. And these types of molecules, catechol molecules, are known to reduce iron in solution. So we asked two questions. One, will it reduce the iron on the surface? And two, will that enhance the decomposition of hydrogen peroxide and enhance the heterogeneous Fenton reaction? And so we started looking at that. And what we have here is the absorption of gallic acid on the surface of the iron oxide clay. So this is what the iron oxide clay looks like. It has an orange hue because of the nano-sized iron oxides. And when we add gallic acid, then it becomes black. So two things are happening here. One, as gallic acid is absorbing, it is polymerizing on the surface, and we could see trimers and dimers coming up in the LCMS analysis. I don't have it here, you'll have to believe me. And the other thing that's happening is that we get reduction of iron on the surface, which also changes the color of the clay. So as we increase the absorbed amount of gallic acid, we're increasing the amount of divalent iron on the surface. And this in turn causes increased decomposition of hydrogen peroxide. So this is the decomposition of hydrogen peroxide on the surface without gallic acid. And this is with two concentrations, low concentration and high concentration of gallic acid. And you can see that there's a direct correlation between, this should be H2O2 decomposition, or as a function of divalent iron. So we were able to reduce the iron on the surface and induce more decomposition of hydrogen peroxide. Why is this advantageous? So there's a lot of wastewater streams that are very rich in polyphenols. Wastewater streams from olive oil manufacturing or from wine or grape juice manufacturing have a lot of polyphenols. Many polyphenols are also catechol. So we wanted to see whether we can use this system to degrade polyphenols on the surface of our catalyst. So we took a bunch of polyphenols that you might find in wastewater from olive oil. This is all synthetic. And you see that some of them have a catechol group, like CC and DHB, and others have similar functional groups, but they're not exactly catechols. So here we have a hydroxyl group and a methoxy group, just methoxy groups, and just a carboxyl group. So the first thing we looked at was absorption and whether or not this reduces the iron on the surface. So you see here absorption isotherms of all 5 compounds, and the 2 compounds that have catechols have kind of a Langmuir-shaped isotherm. This means that they have a specific chemisorption type of absorption, whereas the others have a more linear type absorption trend, which is more indicative of van der Waals forces, hydrogen bonds, and so on. But more importantly, you can see here that only the 2 catechols were able to reduce the iron on the surface, whereas the other ones did not change the speciation of the iron. And when we add hydrogen peroxide into the mix, you can see the decomposition of these compounds. I will say that if you leave them for 24 hours with a catalyst, they will all be degraded. And the TOC goes down to 0. But if you look at the time, if you look at just the first 2 hours, you can see a huge difference between the 2 catechols that are degraded very rapidly as compared to the other 3 that are not catechols. So catechols do induce heterogeneous Fenton. And again, here we did a lot of work on which radicals are more prominent and who does the work. I'm not going to go into all of that, just to show the last experiment that we did, where we took TCA, which was the least reactive compound, and we looked at the degradation of TCA in the presence of each of the other molecules. And that's what you see here. So the black line here is the degradation rate of TCA alone. And then you have the degradation rates with the other 2 non-catechol groups. And then you have the degradation of TCA in the presence of the catechols. So when you add catechols, you get faster degradation of less or more persistent, more recalcitrant pollutants. So you can use catechols to enhance the oxidation of other pollutants. And again, the thought is that the catechols will sit on the surface, reduce the iron, induce the formation of radicals, and some of the radicals can leave that boundary layer and attack the molecules that are in solution. So our next step was to try and tackle more persistent pollutants, because polyphenols are a problem, but they are not considered recalcitrant pollutants. They're not very hard to degrade. They're also biodegradable. We wanted to look at pollutants that are more difficult to degrade in conventional methods. And our idea was, let's bring those pollutants close to the surface. If we bring them close enough to the surface, then similar to what we have with an active site of a protein, everything will happen in the same place. When we add the hydrogen peroxide, then it will be activated on the surface where the pollutant already is, and it will break it down more efficiently. And our overall thinking was that we can create a sorbent catalyst. So if you can imagine a column with a sorbent that absorbs very well your pollutants, and once that column is saturated, you add hydrogen peroxide, which cleans up or regenerates your absorbent in situ or on site without having to burn it or send it somewhere else to be cleaned. So that was kind of the idea. And we chose a specific polymer, this copolymer that you see here, that has a backbone of decafluorinated biphenyl groups that have a kind of hydrophobic characteristic. and beta-cyclodextrins, which are these glucose cups that are known to kind of create a interesting area for pollutants to sit in and kind of be stabilized in. And this has also been shown to work well in Fenton oxidation or other oxidation processes because they get trapped in this cup and then you can degrade them more easily. So we made this polymer, we synthesized this polymer, and then what we did was we synthesized it between dispersed particles of our catalyst. So now we have a composite that is hydrophobic with this catalyst embedded within, with the idea again that the pollutants will sit near the surface, and when we add hydrogen peroxide they will be degraded and they will and that will regenerate that surface. So that was kind of the premise. And initially we started working with three different model pollutants. Bisphenol A, which is a plastic additive, I think most people are familiar with. Carbamazepine is a pharmaceutical, also very recalcitrant, doesn't break down in wastewater treatment plants. We can find it's used as a marker for anthropogenic activities. And perfluorinated compounds. Are you familiar with perfluorinated compounds? Okay, so I'll give a little bit of an overview on these compounds in a minute, but as you can see here, they're fluorinated compounds, very recalcitrant, toxic to humans, and very problematic. And so we looked at the beginning at the absorption to our composite. We broke down the composite to the different components. So we have absorption just to the clay, to the catalyst with just the beta-cyclodextrin cups, to just the backbone of the polymer, the deca-biphenyl fluorinated groups, and then to the whole composite together. And you can see that there's an additive effect for both these two compounds and the fluorinated compound here. So they absorb very well, over 90%. And when we add hydrogen peroxide, they degrade quite rapidly. And these two here, the difference between these two lines here is with and without the beta-cyclodextrin cups. So they add, they are very significant in terms of the degradation. So for example, for the perfluorinated compounds, they weren't very significant for the absorption, but they did help with the degradation. So we have this composite here, which seemed to work quite well. And we're very excited, especially concerning the perfluorinated compounds. So perfluorinated compounds are a really wide family of pollutants. They're what we know as Teflon, used for nonstick pans, used for our Gore-Tex clothes. They've been used for many years as wrappers for food. Have any of you seen the movie Dark Waters with Mark Ruffalo? So yeah. I recommend just to see the movie and then throw away all your pots and pans. So we now know that they're very toxic to humans. They have a negatively charged head group and a long tail or depending on the molecule, that's fluorinated, so both amphiphilic, hydrophobic, and have this negatively charged head. And because they have that negatively charged head, they are very mobile in the environment. And we have research from the last couple of years showing that they're so prevalent and so pervasive, and because they're so persistent and they don't break down in the environment, that they're now in the hydrological cycle and coming down with the rain. And I spoke earlier today with somebody from Rishut Mayim, and they said that now they know that they're also high concentrations in water reservoirs, and we use this water for agriculture. And there's many wells across the country that are no longer good for production water because since the 1st of January 2026, new regulation has come into force, and we now know that they're at very high concentrations in well water as well. So they're a very big problem, and there's no good solution because they don't absorb very well, not to activated carbon, and so there's no conventional treatment method at the moment to take care of them. So we were excited when perfluorinated compounds were absorbed and degraded by our composite, and so we dedicated a whole research project just to perfluorinated compounds. And you can see here that we have 7 different compounds with different chain lengths, and we have one with a sulfonic head group. The most prevalent that you find in wastewater are, in groundwater and wastewater everywhere is PFOS, the 8 carbon with a sulfonate group, PFOA, which is 8 carbons with a carboxylic group, and the small ones. And so you can see here the adsorption to our composite and the degradation after adsorption. So this is not at the same time, right? We adsorb, take out the water, and then we add hydrogen peroxide. So what we can see here is that if the compound is 7 carbons or higher, then it adsorbs very well, and it degrades quite nicely. I mean, 70% degradation is good for perfluorinated compounds. It's fantastic for perfluorinated compounds. But the short ones don't adsorb. And so even if we have a small amount that's absorbed and degraded, we still get most of the bulk of these short ones, they come out of the column or our batch experiments. So the ones that do absorb get degraded. You can see the degradation trends here. This is the amount of fluoride ions that are released. This gives us a proxy for how much is mineralized. So it's not enough just to degrade. You want to turn it into something that's benign. So you need to get rid of all the fluorine. So for PFOA, we get about 70% mineralization out of the 70% that was degraded. So that's quite nice, but for PFOS, it's only around 30%. So still not fantastic, and we're working on this. But what we were most concerned about were the small ones that are not being absorbed. So we turned our attention to trying to increase the affinity of our adsorbent towards the small ones. And so we looked at several different clays, and we looked at several different polymers to put. The idea was that because it's small and negatively charged, we should go towards something more like an ion exchange column. So we looked at positively charged polymers. This PVP coaster, and you can see it has kind of a hydrophobic area here with the styrene group, and then the polyvinyl can be positively charged. And the other polymer is polydabmax, so more hydrophilic and more positively charged. And you can see that the polydabmax gave higher absorption percentage, so about 80% was absorbed, much better than what we saw before. And out of all 3 clays that we used, the montmorillonite that has also the highest surface area was the best. So we worked with the polydabmax montmorillonite composite, and we looked at the absorption. So this is the absorption of PFBA. As I said, it's quite nice, but compared to the other longer ones, PFOA and PFOS, it's still significantly lower. So we can get all of them out with this polydabmax clay composite, the long ones and the shorter ones. And you can see here, this is a mixture of all 3 together at different concentrations. These are quite high. You usually find nanograms per liter in the environment, but you can see that as we increase the concentrations, we're increasing the absorption of the smaller one, which means that we're priming the surface, making it more advantageous. What we did find as a problem was, because we're now looking at electrostatic interactions between the negatively charged PFBA and the positively charged surface, if we add background cations, then they compete with the PFBA, and you get a significant reduction in absorption. So just adding salt decreases the absorption. So if we're looking at real water, we're going to have a problem. So we took some real groundwater from a production well near Yokneam, and you can see here the value of the initial PFBA concentration. It's a well that's an anomaly with just PFBA there. They're not sure where that comes from, maybe from Sultan. They used to have a factory there. But we wanted to see whether our clay with the polymer can, first of all, remove high quantities of this PFBA from water. And you can see here, this is a column experiment. This is the amount of pore volumes. Pore volume is the pores that it goes. So it's a proxy for the amount of volume of water that we passed. And you can see here that throughout the experiment, we used out 4 or 5 liters of water, and we still couldn't get to the regulation limit. This is the new regulation that's come into force now. So it does pretty well, even in real water. But when we added the iron oxides and tried to degrade the PFBA, you can see that the degradation was completely quenched. So with this polymer, we're not getting any degradation of the PFBA. So we changed the polymer. The polymer is now not intercalated between the layers, but it's absorbed on the surface, and that completely quenched our activity. So we can get now good removal, but we can't get degradation. So we're still working on this, and we're working together with Mikorot and trying to think about ways to solve this problem here, and hopefully we'll get somewhere soon. How are we doing for time? We're okay. Okay, so now I want to tell you a little bit about our other directions where we look at absorption and biodegradation. I'll take a sip of water first so you can think about everything I talked about. Okay, so this work was done quite early on when I just started the lab. Door Chemicals came to us. They produce resins that are formaldehyde based, and they had problems with their formaldehyde-rich wastewater. They produce a lot of wastewater that's very rich with formaldehyde, so very high concentrations, 9,000 ppm of formaldehyde, and that wastewater also has a very low pH, and it's very difficult to treat. And so we try to think of a way to degrade formaldehyde from wastewater, and we knew that there were bacteria that know how to degrade formaldehyde to CO2 and water, but they can't tackle such high concentrations of formaldehyde. So the idea was to create a platform where we can stick the bacteria and help them degrade the formaldehyde under better conditions. And we chose a clay and polymer that has a few functions. First of all, that clay polymer composite, I'll show you in a minute the polymer itself, can absorb formaldehyde rapidly. The clay itself mediates the pH, buffers the pH around the bacteria, increasing the pH to around 7 or 8. And then through slow release, the bacteria can degrade the formaldehyde, regenerating the surface for the next batch. So that was kind of the idea. These are a few words about formaldehyde, used a lot in industry. And I've already said this, but bacteria can degrade formaldehyde to CO2, but the conditions in the wastewater are just not bacteria friendly. Okay, so the idea was to take a polymer that had amine groups that can covalently bond formaldehyde, covalently but reversibly depending on pH. So at high pH, we get covalently bonded formaldehyde, and at low pH, it can be released. So you can see here the absorption of formaldehyde to our composite, the clay with the polymer, under different pHs. At high pH, we get high absorption, low pH, we get low absorption. And this is the percent released. At low pH, we get high release, and at high pH, we get a low release. Okay, about 5 to 10%. So,
did you say the wastewater was acidic, so it did not absorb?
The wastewater was acidic, so the bacteria can't live there.
Yeah, but what if it will not absorb to the polymer?
Right, but the polymer with the clay buffers the pH and raises it. I'll show you. Because when montmorillonite has a very strong buffering capacity, and also the clay, and also the polymer. Yeah, this polymer here with amine groups really increases the pH. Okay, and so these two agents together, they raise the pH of the wastewater. We looked at different loadings of the polymer on the clay. We wanted to get the most amount of polymer on the clay. We did that by raising the pH when we created the composite. When you have very high pH, then you also have very strong negative charge on the clay. And the polymer becomes less positively charged, and then it kind of coils in. So you get maximum absorption. It's not spread out, it's coiled, and so you get high quantities of PEI on the surface of the clay, as we see here. And the zeta potential, the charge of the clay, goes from negative to positive, because we added a lot of positively charged polymer. The pKa is around 11 of the polymer. And then we wanted to add bacteria. We took bacteria from an area in the south where they have been treating agricultural fields with formaldehyde as a fungicide. And they contacted a professor in biotechnology, Ayelet Fishman, who was working with me on the project, and they said the formaldehyde is no longer working. She said it's because bacteria have evolved to degrade it. So we took the bacteria from this soil here, and we isolated it. We looked at several different strains. The strains that worked best were Pseudomonas fetida, and we chose the fastest, most resilient strain that we could find, and we saw what the boundaries of the activity of this bacteria was. So we have the OD, so how much they grow under different pHs, and how much they grew under different concentrations of formaldehyde. So you can see that they feel most comfortable around pH 8, the bacteria, and they can degrade 500, maybe 1,000 ppm of formaldehyde, but not higher. So they can do the job, but the wastewater will kill them. The next thing that we checked was the kinetics. So we looked at the kinetics of degradation of formaldehyde, of the bacteria, versus the absorption of formaldehyde to the surface. Because if we want to detoxify the solution and increase the pH without killing the bacteria, we need to lower the concentration of formaldehyde quickly, and then the degradation can take place at a slower step. So you can see here that the absorption is much faster than the degradation. So check on that point. And then we added the bacteria to the surface of the clay. This is the zeta potential of our clay. It starts out positively charged. As we add bacteria, the membrane of bacteria are negatively charged. As we add bacteria, we are reducing the zeta potential. And we chose this amount so we don't get complete coverage of all our amine groups, because some of them, the amine groups are the ones that are captured because they're the positively charged groups. We don't want them all to be taken up because they are also what's capturing the formaldehyde. So we didn't want to oversaturate the surface. This is an SEM. You can see the bacteria here sitting quite nicely nestled within. These are the, this is the clay polymer composite, these things that looks like cornflakes, and these are the bacteria. And then we tested it. So we have a system where we spike formaldehyde. We have a control with just bacteria in suspension. It's not shown here, it was just a straight line. And then we have immobilized non-degrading bacteria, so bacteria that do not know how to degrade formaldehyde, and our complete composite with the FA degrading bacteria, or formaldehyde degrading bacteria. So we added formaldehyde, tracked until depleted, waited for a while to let them eat up what's on the surface, and then repeated for several, as you can see here, consecutive cycles. So at the end of the experiment, the free cells, the wastewater, or the simulated wastewater that had a very low pH, the pH remained low, and the amount of formaldehyde removed was very little, probably just bioaccumulation. And then we have the fixed control, so cells that can't degrade formaldehyde, they can absorb a little bit in the beginning, but then that's it. And then we have our fixed cells, and you can see here that in the blue, we add formaldehyde, you can see that the reduction in formaldehyde in the solution, and then we spike with more, and the reduction again, and we spike with more, reduction again. So overall, over the 10 cycles, we got degradation of over 8,000 ppm. So that was quite nice, and there was a follow-up project that we did trying to upscale it, and a patent was written, and so far nobody's used it. So, the last project that I wanted to show today is something that we did more recently, and this is a little bit off topic, but again, kind of thinking of encapsulation of bacteria to ameliorate their surroundings. So we've been working on designing liquid core alginate beads, meaning the inside is liquid, so just a solution, and the outside is a thin alginate membrane, and the pores are tunable, and so far we've tuned them so that solutes, small molecules, can diffuse in and out quite easily, but the bacteria inside cannot get out, and the enzymes that they secrete cannot get out, and bacteria from the outside can't get in to eat them. So this protects the bacteria inside from predation, and it allows fast diffusion into the capsule. So unlike alginate beads that have been used for many, many years where it's a gel throughout, here we have just a very thin membrane. And so we're thinking this is a high untapped potential for colloidal systems in general. And I'll show you what we've done with this so far, but we have a few ongoing projects as well. So this is what the liquid core bead looks like, and you can see here that we can play around with how thick or thin the shell membrane actually is by using different cations that cross-link the alginate, and then you can get different properties. We've also played around with adding clays on the outside, and then it becomes opaque, and the diffusion is slower, so you can really kind of play around. So what we've done with this so far that's been published is the use of this liquid core beads for encapsulating biogenic manganese oxide for degradation of glyphosate, which is a herbicide. So we chose glyphosate because we knew that manganese oxide can oxidize glyphosate. It's been known from the literature. This is abiotic manganese oxide crystal, and we know that glyphosate can absorb, degrade, and when it degrades, the pathways were pretty much known. This is the degradation on manganese oxides. So you can see this is the pathway. You get phosphate, formic acid, glycine, so mostly things that bacteria can use. And the problem is that manganese oxide, when it oxidizes glyphosate, then you get the release of divalent manganese, and so you lose your surface over time. So we wanted to use biogenic manganese oxide. So these are bacteria that know how to take divalent manganese and create manganese oxide on the surface. First of all, biogenic manganese oxide is known to be more reactive because it's amorphous. So that's one advantage. But the other thing is that once it oxidizes something and divalent manganese is released into the solution, bacteria can take it and regenerate the surface again. So again, we have kind of a regenerable system. So the first question was, does glyphosate degrade on biogenic manganese the same as it does on abiotic manganese oxide? And the answer is yes. And what we found was that as we increase the amount of biogenic manganese oxide, we get faster degradation times, which is intuitive. And the second thing we found was that as we increase the concentration of glyphosate on the same concentration of manganese oxide, then you slow down the reaction. So the reaction rate depends on the ratio between glyphosate to manganese oxide surface area. If you have less manganese per more surface area, faster degradation rates. Okay? So the idea was we can put these bacteria inside our liquid core beads, and then we can have the diffusion, kind of pipette glyphosate in, and then glyphosate meets the manganese oxide surface at a slower rate, or the ratio remains high. And so we put the bacteria inside our liquid core beads. You can see them here. And we tested the rate of degradation. So this is encapsulated bacteria versus suspended bacteria. So essentially these two systems, right? Encapsulated versus suspended, and then we add the glyphosate. And you can see here that at least for the first several cycles, the rate of degradation of glyphosate for the encapsulated bacteria was faster than in suspension. Just because we are controlling, in a way, the ratio between glyphosate and the surface. We have several more projects with these liquid core beads. We want to use them for soil remediation. We want to use them for carbon sequestration. So we have a lot of different ideas. But with that, I'm going to finish for today, but just say that overall, our vision is that adsorption and degradation platforms have the potential to comprehensively treat polluted waters on site. So that's one of the issues that we're trying to tackle. And in general, benign clay minerals and oxides are very promising materials for environmental remediation platforms. And with that, I want to say thank you to all the group. These are the people that actually did the work. I just get to work around with the graphics and the talks. And to you for listening. Thank you.
Thank you. Questions? Okay, so I have one question. So you talked initially about iron oxides and possibly iron oxides on the surface. Have you thought, I know work's been done on zero-valent iron. How does these kinds of things compare with zero-valent iron?
So I think zero-valent iron is better when we want to talk about reduction processes, which may be very advantageous for perfluorinated compounds. So we do have some ideas about working with zero-valent iron, and one of the drawbacks of working with zero-valent iron is, and I didn't mention this, this is in general with nanoparticles, is the agglomeration, right? How do you use nanoparticles in solution? They tend to agglomerate or aggregate, and the clay takes care of that, because now we have them on a surface. So it's definitely one of the things that we have been thinking about, and I think it has potential to be to work, but cooperative.
Okay.
We need people, so yeah. There's a lot of different good ideas that can be,
So the work with the iron oxide was all in oxidized on oxy conditions, because I remember reading some papers about natural environment that have redox changing, and when it's going from anaerobic to aerobic, so we have so this kind of phantom reactions naturally oxidizing sort of organic matter.
because you have divalent iron all of a sudden being oxidized.
Yeah. So can something like this be used for treatment?
So in situ chemical oxidation is something that people have been using for treatment. The problem always with in situ chemical oxidation in soil is heterogeneity of the soil. So where you would have a redox pocket in one place, you won't have it in another place, and then you don't have reduced, it's not, how do you say, you don't get the same results again and again. So we keep getting different results. So usually for in situ chemical oxidation, what they do is they drill a hole and they put in divalent iron and hydrogen peroxide and hope for the best. Sometimes it works very well, other times you get reoccurrence of the pollution. So maybe our problem with this type of technology will be how do we spread the iron clay in the soil evenly so that it will get to all the places where the pollution actually is. So how do you envision to engineer a system where you can distribute these particles that are micron in size in polluted soils? That's one of the difficulties. I don't tackle that difficulty, but if there is a good solution for that, then you'll be able to use this type of technology in soil as well.
Okay, any more questions? Okay, so let's thank our speaker again. Thank you.